Hi, Heather. I am just going to make you a video. I found a little bit of information to help you with your questions about DNA replication versus the single-stranded RNA replication in the coronavirus. So I really like the Khan Academy. I know it's used in high schools as well as colleges, but I think it's a really good resource for basic biology. And DNA replication was covered in Bio 111. So just for a review, I found this information from the Khan Academy. So if you go to the worksheet, it's actually on page 1 of the worksheet in part 1, infection. So this information is going to come from the infection video, but I'm just giving you a little bit of extra information so you can remember about how DNA replication works. So question number 4 says draw and label a model that shows how complementary base pairing is used to create a new strand of DNA during cellular replication. And then it gives you some terms that you need to use in your model. So this is really good for that question right there, because you can copy one of these models if you want to. So just for a review, DNA or deoxyribonucleic acid is the the molecule that contains our genetic information, our genetic code. Nucleotides are the building blocks of DNA, and they're also the building blocks of RNA. So keep your answers to the similarities and differences very simple. For example, both DNA and RNA contain nucleotides, but there is a difference. RNA contains a nucleotide called uracil, and DNA contains a nucleotide called T or thymine. So that's one of the differences there. And it may be something you already know, but like you said, you might have been making it harder than what it was, because there's a large space on that worksheet to write your answers, and the answers are really not that long. So that might have been confusing. DNA is shaped like a double helix, which is a ladder that has been twisted. So it's double-stranded, whereas RNA is single-stranded. At least the RNA in the coronavirus is single-stranded. And DNA replication is the process that occurs in our cells, in eukaryotic cells, including human cells, where double-stranded DNA is copied to produce 2 identical DNA molecules. And base pairing is the principle in which the bases of the DNA bond with one another. Okay, so DNA is a nucleic acid. It is composed of nucleotides, and the nucleotides contain one of 4 different bases, either A, G, C, or T. And remember that when we take the template DNA and make a copy of it, we have to know which bases will pair with which bases, and that comes from Chargaff's rules. And Chargaff found out that the A's always pair with T's and the C's always pair with G's. Again, this is a structure of DNA, the double helix, and it shows you that the nucleotide bases form the rungs of the ladder, or the steps of the ladder, and then the sides of the ladder are composed of sugars and phosphates. But we're mostly concerned with the steps of the ladder. That's where the bases are, and that's what we're concerned with when we talk about replication. So here's where you can use this next information for your model. In DNA replication, you start out with 2. The gray strands represent the original or old strands of DNA, and they separate. And then so in RNA replication, one of the differences is going to be this separation process doesn't occur because there's only a single strand of the RNA. All right, but when the old strands separate, the replication that occurs creates a new strand to bond with each of the old strands. So now you have 2 double-stranded DNA molecules, And we call this process semi-conservative because one of the strands is from the old DNA, and then one of the strands is new that was formed when the base pairing process occurred. Okay, so this is an image of the replication process, and you can copy part of this image for your model that you're supposed to use, because you have to use the terms template strand. So, for example, up here, this is a template strand, and it's got the nucleotides, the bases A, G, T, T, A, G. And what's going to happen is, let's see what else. You have to have the new strand, you have to have base pairs. So RNA polymerase is an enzyme that's going to come and add bases that are complementary to the old strand. So we know that C will go here because it's complementary to G, T is complementary to A is complementary to T, A is complementary to T, and the next one down here would be a C and then a T. So that occurs through an enzyme called DNA polymerase. And in the replication of RNA, the enzyme is called RNA polymerase. So there's a difference right there. And I think I've given you enough information on DNA replication. You don't have to go into a whole lot of detail. You just have to understand some of the basic differences. And if you'll go back and watch the first video on infection, that should give you enough information to compare the RNA replication to the DNA replication. If you only have one similarity and like one or two similarities, one or two differences, you will be fine on that worksheet. Now, if this doesn't help you, if this is not enough information and you still can't get it, please let me know. But I think that this will take you in the right direction.